dive right in. Thank you so much, Harriet and Marcus, for your kind invitation. I promise I'll go back in person and hopefully with less technical issues. So, as all of you know, probably even better than me, melanoma is the fifth most common cancer in men, the sixth in women. And can you please keep clicking? I'm going to go fast through these first slides because it's just background. As you know, the incidence of melanoma continues to increase. It doubles every 20 years. And luckily, this slide, unfortunately, is not updated. The mortality associated with melanoma is starting to decrease. And this is due definitely to the development of better therapies. For the past 15 years, melanoma has become the poster child for many targeted therapies as well as immunotherapies. Please click in. But all of you know that still, despite the success of these ever-developing immunotherapy treatments, we still, can you keep clicking, we are still facing significant resistance to some of these treatments or a severe side effects. So approximately 40% of the metastatic melanoma patients are not benefiting from the advances that have been done over the past years in the treatment of melanoma. So this takes me, can you please go on to the focus of my lab. Can you go back? Yeah, which evolves around these questions, right? The fact that we still need better markers for patient selection, understand better which are the patients that are going to go on to recur after the initial resection of their tumor, which ones are going to metastasize. And also, in particular, as Harriet mentioned, we've developed an interest in understanding the biology of CNS metastases, particularly of brain metastases, because they show less durable responses to immunotherapy, and their incidence is increasing as patients are living longer and surviving from other systemic metastases. So my lab is focused in two aspects of metastasis: the initial steps of early dissemination. We now know that primary tumors, especially in melanoma, start disseminating, start shedding cells that go into circulation and metastasize really early on in the process. So we want to understand, please click, which are the mechanisms that drive the metastatic behavior of certain melanomas and not others. We know that genetic alterations don't explain this behavior because none of the well-characterized genetic alterations of melanoma or combinations of those genetic alterations can explain why some primary tumors recur and metastasize and some others don't. And also we want to understand what is the contribution of intratumor heterogeneity. So we know now that despite the fact that primary tumors can have a relatively homogeneous genetic profile, inside those tumors we can recognize groups of cells with different transcriptional programs, what we call transcriptional states. And there is a recent search for what are the programs or the states that drive this metastatic behavior. And people have pointed to the neural crest-like or the EMT program. On the other side of the equation, we are also trying to understand what happens at the end of the process. Once the cancer cells, in this case melanoma cells, have already extravasated into those distal organs. As you know, melanoma metastasizes to the lung, the liver, and the brain. And we are interested in understanding what are the site-specific adaptations in those sites. What are the type of metabolic changes, the cell types that the cancer cells are interacting in, what is this crosstalk between the cancer cells and their environment, how is shaping the ability of melanoma cells to adapt and grow in those environments. And I will have two stories presenting today, if time permits. One that focuses more in just the metastatic potential in general and how melanoma cells co-opt some of the programs of the neural crest cells from which they arise. And also in the second part of the talk, I will talk more specifically about brain metastases and how melanoma cells mimic some of the processes that happen during neurodegeneration, particularly to suppress neuroinflammation and be able to grow within the brain. So diving into the first story, these are the 4 areas in which my lab has been focused in recent years: epigenetic alterations, non-coding RNA, post-translational modifications, particularly glycosylation, and site-specific adaptation. So moving on, can you please move the slides? One more. The first story that has been recently accepted for publication is the work of 3 people. It evolved over time, and we have titled it Melanoma Cells Invoke a Neural Crest Epigenetic Program During Metastasis. So the genesis for this project was a very simple premise, and is the fact that, well, we all know that neural crest cells are the cells of origin of melanocytes, and these are among the most totipotent and among the most invasive and migratory cells of our body. And it's well known now, if you keep clicking, that melanoma cells adopt programs characteristic of neural crest cells during the progression from primary to metastatic cancer. So we wanted to investigate which epigenetic changes happen during melanocyte differentiation from the neural crest to the melanocyte that potentially could be reversed in the transition from primary to metastasis. And we did this by looking specifically at DNA methylation. There are, of course, many other mechanisms of epigenetic regulation, being DNA methylation one of them. So if you move to the next slide, please. What we did basically was a comparison of 4 data sets. So in one hand, I cannot use my pointer, but you can see that we have 4 columns. We have neural crest cells that were obtained from a collaborator in France. These are human neural crest cells. We also had the human melanocytes from different donors. And as you can see, there is kind of a mirroring pattern. We were looking for CpG islands that were either hypomethylated in the conversion from neural crest cells to melanocytes that were progressively hypermethylated from primary to metastatic melanoma, or the converse. If you see at the bottom, we have some CpG islands. If you can click again, you will see there are some CpG islands around the gene called NR2F2 that are hypomethylated in the neural crest cells that become hypermethylated in melanocytes and progressively hypomethylated from primary to metastatic melanoma. And this really caught our attention because it really represented an example of a potential gene or candidate program to be modulated during neural crest differentiation to melanocytes that was reversed during the progression from primary to metastatic melanoma. And what is, first of all, NR2F2? So it's a nuclear receptor, is also called CUPTF2, is an orphan nuclear receptor. We don't know still what is the natural ligand, although retinoic acid can bind it at high concentrations. This is the motif that it binds, and as you can see, the NR2F2 full isoform has a DNA binding domain, it has a ligand domain, so it's a conventional nuclear receptor that can form homo or heterodimers. It is essential if you, it's essential for development and particularly for the formation of blood vessels. And it has been shown to be important for neural crest differentiation. Now, there had been already some studies showing the role of NR2F2 in cancer, particularly pancreatic and prostate cancer metastasis, whereas in breast cancer, it had controversial effects. So you can say, well, you know, this may already be known that this factor is important for cancer. However, if you click one more slide, what really called our attention is that the CpG islands that we found to be differentially methylated in neural crest cells versus melanocytes, and then later on in primary versus melanoma, were actually affecting exactly that region in the transcription start site that controls the expression of an alternative isoform, isoform 2, which is not the one that has been commonly characterized. So most studies have focused on the full-length isoform, isoform 1, that has the DNA binding domain, the ligand binding domain, but this methylated region or demethylated region is in a TSS that gives rise to a truncated isoform that lacks the DNA binding domain. So on the right, you can see that as we had shown before, the CpG islands controlling this nuclear receptor, NR2F2 iso2, are hypomethylated and they appear in green. in neural crest cells and embryonic stem cells, but completely hypermethylated in melanocytes. And this is also described here at the bottom, where you can see the beta value. A beta value of 1 means completely methylated, and a beta value close to 0 represents a hypomethylated gene. This completely corresponds to gene expression, because you can see that whereas isoform 1 is expressed in ES cells, neural crest cells, and melanocytes, isoform 2 is expressed in ES cells and neural crest cells, but not expressed at all in melanocytes. Okay, so when you look at human samples, you can see that even though it's not completely black and white, what we see is that there is an increased percentage of hypomethylation of ISO2 in the metastatic cases compared to the primary cases. You can see there is from 30% approximately to more than 50% of the samples have hypomethylation for NR2F2 ISO2. And if you keep clicking, you will see that you can see there is a progressive increase in the beta value from nevi to primary metastasis, so more hypomethylated, and conversely increased expression from primary to metastasis with a very nice correlation between mRNA expression and methylation. Please, exactly. If you look at the protein levels, we were able to develop actually an antibody specific for isoform 2, which was challenging because there are only 15 amino acids in the amino terminal that are specific for ISO2. You can see that there is an increase in the expression of isoform 2. There are few cells that are positive in the primary cases, and this population expands in the metastasis. This is something that we are actually trying to understand now, whether there is a selection for those cells that express the isoform 2 over time. Again, this is something we're currently trying to investigate. But if we can go on, we can see that isoform 1, the one that is the full length isoform, is not modulated by methylation, is completely hypomethylated in ES cells, neural crest cells, melanocytes, and both in primary and metastatic melanoma. And there is no correlation between expression and methylation. So isoform 1 is always there, but it's isoform 2, the one that is normally not expressed in melanocytes, but is increasingly demethylated and expressed from primary to metastatic melanoma. Now, of course, the big question is this isoform 2 doing anything in melanoma metastasis? And for that, we had to move to cell lines. And again, we found this pattern in which melanocytes and some melanoma cell lines had complete hypermethylation and lack of expression, as you can see here on the right. And you can also see a hypomethylation in some of the cell lines, and this corresponds to expression. And the same can be seen at the level of protein. You can see isoform 2 expressed only in those cell lines that have hypomethylation. So this represents a good model to study loss of function and gain of function. But before I go into that, you can see that the methylation status can also be seen in short-term culture. These are cells isolated from patients, and again, you have some of these short-term cultures have hypomethylation, and some of them have hypermethylation. So this is not an artifact of in vitro culturing. It happens in cells derived very shortly from patients. So if now we move into cells that have hypermethylation, like Mio cells, if we treat them with a demethylating agent like 5-ESA, you can see that there is an induction of isoform 2, and this is seen also in short-term cultures in which treatment with 5-ESA results in induction of isoform 2 expression with no change in isoform 1. Now, when we silence isoform 2 using specific shRNAs against this isoform that don't have any impact on the other isoform, initially we did not see any effect onto the proliferation, but we observed a clear decrease in the ability to form colonies in soft agar or to form spheres upon single cells. So these are properties that are characteristic of metastatic cells, and basically measure the ability of the cells to grow under very stressful conditions. Now, if we overexpressed, before we go into the overexpression, this is the in vivo experiment. In this case, we injected the cancer cells intracardiac in a model of metastasis that we use very often in the lab. And you can see that the silencing of isoform 2 with two independent shRNAs had a very significant decrease in metastatic potential in this model, which is quantified here on the right by bioluminescence as well as fluorescence intensity when we extract the organs. So it seems like isoform 2 silencing suppresses metastasis in this model. And now we move into overexpression systems in which ectopic expression of isoform 2 in cells that have hypermethylation has no effect again in 2D culture, but it has a significant ability to increase the number of colonies and soft agar as well as sphere formation. In vivo, we injected these cells in the flank of the mice, and then we did survival surgery. You can see that we cover the tumor area because there is always a lot of signal that comes even after you have resected the majority of the tumor, the subcutaneous tumor, and you can see that ISO2 overexpression enhances metastasis both by bioluminescence as well as by histological analysis. We did this also by intracardiac injection, and again, we observed the same effect with an increase in metastasis overall. So it seems like ISO2, this particular truncated isoform of the orphan nuclear receptor NR2F2 is able to promote metastasis, and it seems to be required in some models in most of the models that we tried in lab. In the paper, we have 3 or 4 different models. You can see a decrease in metastasis when you silence or knock out this gene. So now, what are the programs that are modulated by isoform 2? We did RNA sequencing to get to this question. And one of the pathways that was significantly modulated was EMT, the epithelial to mesenchymal transition. Now, we went on to demonstrate that a lot of the typical genes involved in the epithelial to mesenchymal transition were silenced in different cell lines when we deplete isoform 2. And here you can see a validation for snail, which is reduced both transcriptionally and at the protein level when we silence isoform 2. Importantly, as I've mentioned at the beginning of my talk, now we have an understanding of intratumor heterogeneity in melanoma. And we have seen that most melanomas, both in mouse and in human, display different transcriptional states, intermediate, neural crest, like proliferation, EMT, or more melanocytic or differentiated. So we were curious to see whether NR2F2, and particularly the signature of genes modulated by isoform 2 were particularly enriched in any of these transcriptional states. And as you can see in this heat map, the EMT in particular, the transcriptional state that corresponds to an epithelial to mesenchymal transition, seems to be the one that is more enriched in this NR2F2 signature together with the neural crest signature. So this goes together with our findings that NR2F2 could be regulating the EMT state. And in this additional analysis in which we used CINIC to understand the regulons, basically the epigenetic and transcriptional factors that control each of these states, melanocytic, intermediate, proliferative, neural crest-like, and EMT, you can see, and I think is more visible in the next slide, that the top regulon controlling the EMT signature, and this is a completely different analysis done in our mouse models of melanoma. You can see that NR2F2 is at the top, is a top regulon controlling the EMT signature. So it seems like this transcriptional state of epithelial to mesenchymal transition seems to be mostly controlled by NR2F2. Now, how this happens, right? I told you at the beginning that NR2F2 iso2 is a truncated isoform. It lacks the DNA binding domain. So how is it possible that it has this capacity to control gene expression? Now, when we started to work in this isoform, there were only 2 other papers that were studying alternative isoforms of NR2F2. All the papers that have been put out there study only the full-length isoform. So there were 2 papers. One was suggesting that iso2 acted as a dominant negative and was removing or displacing isoform 1 from chromatin. Whereas another paper suggested the opposite, that ISO2 contributed to bind ISO1 to the chromatin. And also what complicates more this interpretation of the results is the fact that isoform 1 has been, or NR2F2 has been described both as a receptor, as a repressor of transcription as well as an activator. So we it tested the possibility that NR2F2 ISO2 was interacting with isoform 1 and somehow modulating the ability of NR2F2 to bind chromatin and regulate gene expression. But for this to be true, isoform 2 had to have the capacity to bind to isoform 1 in the nucleus. And isoform 2 lacks the nuclear localization signal present in isoform 1. It has an alternative nuclear localization signal that is much less potent. So we first show in a fragmentation analysis that isoform 2 is able to reach the nucleus. You can see it there, not only the cytoplasm, right? So this suggests the possibility that it can interact with isoform 1. Then we also did IP analysis where we show that isoform 1 can pull down isoform 2 with 2 independent antibodies, and the opposite is also true. We can use now a GFP pull down because unfortunately the endogenous IP for ISO2 doesn't work well. So we use the exogenous construct that has a GFP fusion. And so we can pull down again isoform 2 with the isoform 1, suggesting that these two isoforms interact. And when we look now at CHIP of isoform or chromatin IP of isoform 1, we can see that indeed when we silence isoform 2, there is a group of peaks of isoform 1 that now lose binding of isoform 1. And this is more dramatic in this group of targets, is a little bit less prevalent in this subset of peaks that are bound by isoform 1. But we can also observe the opposite. We can see that in a group of peaks, there is an increased binding of isoform 1 when ISO2 is gone. So definitely the interaction between these two isoforms is complex. We know that the chip works because NR2F2 is the most significantly enriched transcription factor in these peaks, together with some other transcription factors, which we're also interested in looking at the potential interaction of NR2F2 with those. So we can see that NR2F2 ISO2 modulates the binding capacity of isoform 1 to the chromatin, but it not only in a way seems to be in a gene-specific manner. And when we integrate the chip data with our transcriptomic or RNA sequencing of cells in which we have depleted ISO2, we found that the majority of the genes, or a significant portion of the genes that are downregulated when we silence ISO2, that are targets for NR2F2, are genes that are involved in the EMT, snail, twist, TGFbR2, etc. So it seems like a lot of the NR2F2 ISO1 targets are actually direct EMT genes. The opposite is that when we look at the genes that are now upregulated by isoform 2, the majority of those genes are involved in differentiation and pigmentation, such as tyrosinase, DCT, etc. So with all of these, and still with many questions still open, we propose this model in which in primary tumors, isoform 2 is hypermethylated and silenced, so the dimers of isoform 1 prevail over the heterodimers of isoform 2 and isoform 1, whereas in metastasis, we see a displacement of the equilibrium towards the heterodimers, because now isoform 2 is present. And it's this heterodimer that allows the expression of neural crest and EMT genes, such as twist, SLAG, etc. So what we are now in the process of understanding is how this heterodimer is able to activate these EMT genes. And we think that in part could be due to the interaction with third parties, like additional transcription factors that are specifically attracted to the complex by isoform 2. So we are doing now RIME and other techniques to understand what are the complexes that are attracted and bound to the chromatin when we have the homodimers versus the heterodimers. Now, moving on into the second part of my talk, I don't think I have to repeat the conclusions, because that's basically the summary that I just have provided. So in the interest of time, I will move to the second part of the story, in which we have focused on brain metastasis. This was the work mostly done by and I actually incorrectly stated PhD is an MD PhD from our laboratory, Kevin Klefman, that is now a resident at Mass General. So we, of course, are interested in brain metastasis because it's an important unmet clinical need. And although these tumors can respond to immunotherapy, we know that these responses are mostly seen in patients that are asymptomatic. And in the symptomatic patients, the responses are much poorer, and these patients have overall very poor survival. So this remains a very important clinical question, and definitely a fascinating biological question, how melanoma cells adapt to the brain microenvironment, and why they have such a profound tropism for the brain is still something that we don't entirely understand. So in collaboration with Eva Hernando-Monge, the director of the melanoma program at NYU, we developed melanoma short-term cultures that in some cases are derived from the same patient. So we can, in some of the cases, we were able to obtain short-term cultures derived from a brain metastasis and extracranial metastasis from the same patient. And again, this is a very difficult comparison to make, but we think it's very useful because it reduces some of the intertumor heterogeneity that we observe both genetically and transcriptionally. And what was really exciting to us is that when we label these cells with GFP luciferase and inject them back into mice with intracardiac injections, we observed that metastatic, the short-term culture that has been derived from the brain has in general more metastatic potential than the one that was derived from an extracranial metastasis. This is represented here on the right. But more specifically, the one that derives from the brain has more ability to metastasize to the brain. And this is measured here as a ratio of brain to body luminescence. So it seems like these short-term cultures retain some of the properties, some of the ability that they had gained in vivo in the patients of colonizing the brain, and therefore it could be a good model to study brain-specific adaptations. So we went on to conduct a proteomic analysis of these short-term cultures. In total, we profiled 25, approximately 12 and 13 brain metastases versus extracranial metastases. Only a few of them, of course, were paired, the rest were unpaired. And the idea was to try to identify proteins that were differentially expressed in the brain metastasis and could be potential drivers of the adaptation. The first surprise that we found when analyzing the data is that the majority of the proteins found differentially expressed were proteins involved in neurodegenerative disorders such as Parkinson's, Alzheimer's, OXPHOS. And this was rewarding because our collaborator in MD Anderson, Mike Davis, had previously found that a lot of the proteins involved in a different study, transcriptional profiling of brain metastases, were enriched in OXPHOS or proteins involved in the respiratory chain mitochondria. So this was confirmed in our hands. We found that short-term cultures of brain metastasis had elongated mitochondria, and they also had increased oxygen consumption rate in this seahorse analysis. But what we focused on was in the differential expression of proteins involved in Alzheimer's and Parkinson's disease. In particular, we landed for this study on APP, the amyloid processing protein. That is a precursor for amyloid beta, which was induced in brain metastasis compared to extracranial metastasis, but not only APP itself, but the proteins that cleave APP into amyloid beta, like beta secretase or presenilin. So we decided to modulate the loss of APP to see if it had an effect on brain metastasis. And initially, we found that went too fast. We found that supernatants of brain metastasis-derived short-term cultures had higher secretion of amyloid beta compared to the extracranial brain metastasis, not only in our own hands, but also in short-term cultures obtained from collaborators at the Wistar Institute. We silenced APP. Again, we found no effect in proliferation in culture, but when we inject these cells intracardiac in immunodeficient mice, we observed this reduction of the brain-to-body ratio, suggesting that the loss of APP was particularly affecting brain metastasis. This was confirmed by histological analysis. This is a staining of NUMA, which is a human marker, and therefore it can perfectly mark the cells that are deriving from the xenograft, from the implant. And you can see that there was a very significant reduction of brain NUA positive cells, but no effect on kidney or liver metastasis. We did ex vivo imaging MRI to conduct volumetric analysis that show us that there were not only less brain metastasis, but also smaller brain metastasis. And of course, we show that this effect happens in other models. This is in a braintropic melanoma cell line, 5P1, and we use an orthogonal approach, in this case, CRISPR-Cas9, to show again a reduction in brain metastasis. But this, of course, open a lot of questions. The first one is, which step of brain metastasis is the one in which amyloid beta is particularly required for the adaptation and the arrival of melanoma cells to the brain? So, as you know, brain metastasis is a complex process. It involves multiple steps, the intravasation from the tumor first into the stroma, then the intravasation into the vasculature, survival in circulation, and when the cells arrive to the distal site, in this case the brain, they have to again extravasate. Many of these cells will undergo cell death or become dormant, but those that are able to proliferate and survive in this environment will form micro and macro metastasis. So when was A beta required for this process? So Kevin embarked himself in really a difficult task of monitoring the kinetics of cancer cells injected intracardiac in these mice. So he did brain slice immunofluorescence and a lot of confocal microscopy, and was tracking these GFP positive cells in the brain over days. So you can see that just one day after a intracardiac injection, these cells are stuck in the vasculature. They even have the shape of the blood vessels. This is a tomato lectin marking the blood vessels. You can see that at day 3, they start extravasating. They start getting out of the vasculature. Some of these cells die. A lot of these cells die in the blood vessels or outside when they are able to extravasate, as stained by Cliff Caspase 3. And you can see that later on they start crawling through the blood vessels in this process called vascular cooption. They can later on form micrometastasis at day 14 and finally macrometastasis. So when you compare the kinetics of control cells here in the black line to those that lack APP, you can see that the first steps of the kinetics are really similar. There is this big crisis where most of the cells that are able to extravasate die, either in the vessels or right after extravasation. But then after day 7, when the control cells are able to start expanding and proliferating, happily, the ones that lack APP can no longer grow after the first or second division, and they eventually disappear. They are dead. So we wonder which effects were required for this role of A beta in the brain. And remember that this is a very complex environment where there are, the resident myeloid cells, the microglia, macrophages, in some cases, bone marrow-derived macrophages that are attracted to the tumor, dendritic cells, neutrophils, lymphocytes, and astrocytes. So we first look at astrocytes as a potential a cell of interest because of the literature that has shown previously that activated astrocytes can be co-opted by the cancer cells to support the growth in the brain environment. And this is indeed the case also in our models, where we see that if you look at the left panels, you can see that over time, as the melanoma cells arrive to the brain, these are the control cells, you can see an increased presence of GFAP positive astrocytes. So there is some recruitment of activated astrocytes. We cannot distinguish if it's recruitment versus activation of the surrounding astrocytes, to the point that they form this network of active astrocytes that is called active astrocytosis, supporting the melanoma marker metastasis. So what we observe is that cells that lack APP are unable to trigger this reactive astrocytosis around them when they arrive to the brain. There is a significant reduction of positive cells of GFAP, positive cells around the cells, the melanoma cells that lack APP, suggesting that perhaps A-beta is important in triggering this astrocytosis. Now, in data that I don't have the time to explain, we also show that amyloid beta not only has the capacity to activate the astrocytes, but also it can suppress the phagocytosis coming from the microglia. So we can see that there is a reduction of neuroinflammation in the presence of amyloid beta. So we think that amyloid beta secreted by cancer cells can have multiple effects in the brain metastasis microenvironment, particularly on the astrocytes and the microglia, but also it can influence, as it has been reported in Alzheimer's, the interaction with the endothelial cells. So, of course, these findings open some possibilities and therapeutic opportunities because of all the armamentarium of drugs that have been developed against amyloid beta, some of them beta secretase inhibitors, and more recently, anti-amyloid beta antibodies, some of which have been developed for clinical use and in some cases even approved by the FDA. So these open the possibility of repurposing some of these drugs, which are generally safe, for brain metastasis. And for proof of principle, we've been testing some of these compounds in collaboration with Eli Lilly. So we obtained beta secretase inhibitors in the diet of the mice. So initially we injected cancer cells, melanoma cells in these mice and gave them beta secretase inhibitor in the food or control diet. And you can see how this reduces the number of brain metastases in this model. This is the short-term culture, but also in the 5B1 melanoma cells. Now, of course, this is more a prophylactic model because treatment starts at the time of injection. So we raised the bar a little bit by allowing the cells to establish metastasis first. And then after 21 days, we gave doxycycline to the food and the water of the mice to activate DOX-inducible shRNAs. And again, we saw that in this context, even when the treatment is initiated once metastases have been formed, we can see a reduction in the number of brain metastases. And if we do the same thing with the beta secretase inhibitors, we can also see, again, after initiating the treatment, once the micrometastases have been formed, we can see a reduction on brain metastases. We are currently trying to move into, well, before I go into that, we are now testing the antibodies against amyloid beta, either as a monotherapy or in combination with immunotherapy to see if we can recapitulate the same effects observed here. So to summarize this part of the talk, we have shown the proteomic studies have revealed a novel connection between brain metastasis and neurodegenerative pathologies. This has now been confirmed by other studies. We collaborated with a group of Ben Iser last year. We got a study done in single cell analysis of brain metastasis that also recapitulated this finding that melanoma cells mimic the neuronal pathways and a lot of the alterations that are seen in neurodegenerative disorders once they reach the brain. We see that amyloid beta is particularly required for brain metastasis and not other sites of metastasis. is required for steps that happen after extravasation and early survival in the brain parenchyma. And among the multiple functions of amyloid beta in this context, we have seen that it triggers an anti-inflammatory response in the astrocytes and suppresses neuroinflammation. As I mentioned, we are also studying now whether these effects of amyloid beta can be seen also in other models. So we have done a spatial transcriptomic analysis. In this case, it's not a melanoma model, it's the 41 model, which is a breast cancer triple negative model that also colonizes the brain after intracardiac injection. These are at the top is a brain, is half of a brain of a sham mouse, and this is the mouse. This is a mouse that was injected with the 41 cells. And what I want to bring to your attention is that in this 10x vision analysis, we can see that the GFAP positive cells around the areas where the tumors are. You have to probably take my word. These are the areas where the tumors are, circled here. And we see a special expression or increased expression of GFAP around those tumor cells. We also see S100, which has been seen induced in tumor cells in the brain environment in other cancer types. And interestingly, we find this signature that we have, that has been previously reported as an Alzheimer's-associated microglia signature that is a combination of 10 markers and is again seen particularly activated around the brain metastases here and here. So it seems like perhaps this Alzheimer-like response in the microglia genes around the brain metastasis cells could be a more general finding and not only characteristic of melanoma. As I mentioned, we are now very excited by studying whether APP genetic and pharmacological inhibition extends to other cancer types, and whether the combination of beta secretase inhibitors or amyloid beta antibodies can work alone or in combination with checkpoint blockade in immune competent models. I was hoping to tell you about another very exciting story in the lab, but I see the clock and we are reaching the 1 p.m. So I will stop here. And this is data I wanted to present, but perhaps at the second occasion. And just want to thank all the members of the lab for their contributions. The work that I presented was mostly done by the first part, Richard, Veronica, and Claudia in NR2F2, and Ali and Maya in CNDP1, and Kevin Kleffman led amyloid beta story. And I want to thank, of course, all our funding sources, and thank you all for your attention and your patience with the technical issues at the beginning. I'll stop here and take any questions. Thank you.
All right. Thank you so much, Eva. I'm going to ask folks to put questions in the chat. We only got a couple of minutes, but while people type in their questions, maybe I don't know if Marcus has any, or I can ask one. Go ahead, Marcus.
I've got plenty.
Yeah.
So Eva, really, congrats also on the Nature Communications paper. And I was wondering, with the NR2F2 story, you're probably aware of Chris Marine's work in Nature last fall on TCF4. And I'm kind of wondering if you think your NR2F2 is upstream of TCF4, if you've seen any role there. And there's probably going to be a subsequent story about resistance to therapies. So there's multiple parts to this question. A, the upstream, downstream part, but then also the heterogeneity, because you're talking about these things happening sort of uniformly, but I'm imagining that the epigenetic regulation is kind of cell by cell, and that there's going to be a population of cells that have more or less of that along the way.
Yeah, these are all excellent questions, and these are all the questions that we're trying to address now. So we didn't see a total overlap between our population of NR2F2 with Chris Marine's population. Yeah. It seems like his population is a smaller one. NR2F2 seems to be more broadly expressed in both EMT and also partially in the neural crest-like population. So his population seems to be a more, I wouldn't say minority, but it's a smaller population and not directly overlap. you also, the other challenge that we have is that a lot of the single cell analysis that have allowed us to distinguish these transcriptional states don't have the depth to look into isoforms, right? So you really have to have a different library preparation and pipeline to identify the different isoforms. So when people look at NR2F2, they are just examining NR2F2 ISO1, the full length, and the one that is really switching from the primary to the metastatic, the one that is really triggering the EMT-like program is only isoform 2 because of this balance between homo and heterodimers. So we're definitely trying to understand what is the hierarchy between these pathways, whether NR2F2 is upstream. It seems to be one of the critical regulators. So again, when we do this regular analysis, the scenic analysis, but still we don't know which one is upstream of which, right? That's a very good question. And then you ask the dynamics, right, of these populations. So we are now in the process of labeling endogenously this isoform to be able to trace them. We want to understand whether these isoforms are dynamically regulated during the metastatic process. And we have different labeling systems now that allow us to monitor single cells during metastasis. So I hope we will have soon an answer for that, but we don't know. I also anticipate that it would change from the primary tumor as the cells switch from the more proliferative to the more invasive, and then back when they arrive to the metastatic side, but still we don't have full evidence for that.
So there were a couple of questions in the chat, both relating to Alzheimer's disease and brain metastases. One, is there an increased incidence in brain metastases among Alzheimer's disease patients? And the other one was whether at postmortem you see Alzheimer's plaques in patients with brain metastases.
Great questions. So we haven't seen brain metastasis. We haven't seen plaques in brain metastasis. We look for them. We don't think that they get to accumulate. So we don't think that the processing is wrong. It's just simply induced. So we see more soluble A beta being produced, but we don't see oligomers and we don't see plaques. We did that staining that conventionally is done by pathologists to look at the plaques, and we couldn't see it either in our models nor in human samples. And that's the reason why we think that the antibodies that we want to try are antibodies that are designed against the soluble amyloid beta and not against the plaques, which are the ones that have been now, or the oligomers, which have been now approved by FDA. Now, you asked the other question, the incidence in Alzheimer's. So we look into that, and we didn't see in epidemiological studies an association between Alzheimer's and brain metastasis. But normally, remember that in the majority of the neurodegenerative disorders, there is an inverse correlation between cancer incidence and neurodegenerative disorders, particularly Alzheimer's and Parkinson. And even though there is not a positive correlation, there is no negative association. Now, there is a reported association between Parkinson's and melanoma brain metastasis. I mean, I think Harriet is skeptical, or not. I see you.
No, there's a slight increase, but anyway, I don't know that it's brain metastases specifically. It's just melanoma.
Sorry, it's melanoma. Sorry. Yeah. I misspoke. I meant melanoma in general. Yeah. Mm-hmm.
Yeah. No, but there's a really interesting observation there. So thank you. And thank you so much for this amazing presentation. I have more questions, but I'm going to email them to you. I don't believe there are any more in the chat. We appreciate your patience with all the technical challenges, and thanks for virtually visiting. Next time it'll be in person. Thanks for a wonderful talk too. Really fascinating work.
Thank you, Harriet. Looking forward to see you soon, and thanks everybody. Bye.